Lymphatic tissue mainly deals with the immunity of our body, and it also drains the large particulate matter which is not normally drained by the systemic circulation. So we can define the lymphatic tissue as specialized form of connective tissue that mainly has immune cells, and it also forms supportive framework or meshwork with the help of reticular fibers and reticular cells. And lymphoid tissue identifies foreign antigens, then the dead or old cells of the body, and it removes them by phagocytosis or by producing antibodies. Functions of lymphatic system are: it drains the large particulate matter and colloid component, then it absorbs the digested fats, it filters toxic agents with the help of lymph nodes. So the lymph node helps in filtration of the lymph or the fluid of the lymphatic system, and the toxic agents are removed by the macrophages present in lymph node. Then the lymph follicles produce T lymphocytes and the plasma cells produce antibodies. The T lymphocytes mainly deal with the cellular immunity, and the B lymphocytes produce humoral antibodies. Also, the clinical significance of lymphatic system is that it acts as a channel for distant metastasis. Distant metastasis means spread of infection like tuberculosis or malignancies can spread through lymphatic system. Then the antitoxic serum given for antitoxic or anti-venom serum given in case of snake bite also enters the tissue spaces, and it is absorbed by lymphatics. Lymphatic system protects our body by imparting immunity, and immunity is the ability to fight against infection by producing antibodies. And the immunity is again of two types. Innate means the immunity present at birth, and acquired immunity is the second variety which the individual has throughout his life. Acquired immunity again is of two types, active and passive. Active means when an individual is exposed to some toxic agents or antigen or microorganism, then the immune system gets activated and it gives acquired immunity. And passive immunity means there is already formed antibody and that is transferred to the individual that imparts immunity. So that is passive immunity. So to revise, lymphatic system provides immunity. Immunity is the ability to fight against infection. It is of two types. Innate means at present at birth. Acquired, it is acquired throughout the life. Acquired again is of two types. Active, which means the cells are activated after exposure to antigens, and passive means already antibodies are there. They are transferred like we inject immunoglobulins or we give vaccines. They provide passive immunity. Lymphoid organs are classified as primary lymphoid organs and secondary lymphoid organs. So those lymphoid organs that produce immature lymphocyte from stem cells are known as central lymphoid organ or primary lymphoid organ. So they produce immature lymphocytes. An example is thymus and bone marrow. Whereas secondary lymphoid organs are those organs which when exposed to antigens convert these immature lymphocytes into active lymphocytes. And example of secondary lymphoid organ is spleen, lymph node, tonsil, appendix and Peyer's patches. Histologically, the lymphoid tissue can be grouped as diffuse lymphoid tissue and dense lymphoid tissue. Now, what is the difference between these two? In diffuse lymphoid tissue, the lymphocytes are diffusely distributed throughout the mucosa of the hollow viscera like gastrointestinal tract, respiratory tract, urinary tract. So, as the lymphocytes are diffusely distributed, and they are not segregated or they are not encapsulated, they are known as diffuse lymphoid tissue. An example is Peyer's patches present in the ileum of small intestine. It is an example of diffuse lymphoid tissue. Diffuse lymphoid tissue is also known as MALT, mucosa associated lymphoid tissue, because the lymphatic tissue is distributed over the mucosa. And second variety is dense lymphoid tissue. So here the uh, lymphoid tissue is densely aggregated and it is encapsulated, and it forms a discrete organ. For example, lymph nodes, spleen, thymus, and tonsil. So these are dense lymphoid tissues. Now we will see different cells of the lymphoid system. So the lymphoid system mainly has immunity related cells known as lymphocytes, and as it is specialized variety of connective tissue, it forms supportive framework for various organs with the help of the supporting cells like reticular cells, epithelio-reticular cells. So there are two types of cells we can say related to immunity are lymphocytes, and forming the reticular framework or supportive uh, meshwork is reticular cells and epithelio-reticular cells. Now there are some cells of activated white blood cells which also imparts immunity, or they get activated during immune response. So these are monocytes, macrophages, neutrophils, basophils, and dendritic cells. And initially the lymphoid tissue is arising from the stem cells which is undifferentiated. These are immature lymphocytes, and the cluster of differentiation molecules or the CD molecules will help in the differentiation of this lymphoid tissue. We are seeing the cells of lymphoid system. So the first variety of cell is lymphocyte. Lymphocyte is a subtype of white blood cells, and these lymphocyte, if we see histologically, they have centrally placed round nucleus and there is thin rim of cytoplasm, and the nucleus stains darkly blue because it is heterochromatic. And lymphocytes are again of two types, small size and large size. Depending on their size, small and large lymphocytes are there, and depending on the presence of cluster differentiation molecule, they are further divided into three types: T lymphocytes, B lymphocytes, and natural killer cells. And all these originate from bone marrow. Lymphocytes are grouped into three varieties: T lymphocytes, B lymphocytes, and natural killer cells. So the T lymphocytes are located in the paracortical area of lymph node, periarterial sheath of spleen. Then it is also present diffusely in the circulatory system. Also we can see the T lymphocytes, and the T lymphocytes secrete lymphokines, and these lymphokines are known as interleukins. And the interleukin 4 secreted by T lymphocyte helps to differentiate B cells, and the T cell receptor proteins at the surface recognizes antigen cell mediated immunity. Undifferentiated lymphocytes originate from bone marrow, and after that it enters in the thymus. In the thymus, these undifferentiated lymphocytes will differentiate into immune competent mature T lymphocytes, and it will maintain the cell mediated immunity. Based on the presence of specific cluster differentiation antigen, the T lymphocytes are divided into three groups. Helper T cells are known as CD4 cells, cytotoxic T cells are known as CD8 cells, and the suppressor cells are the regulatory T cells. The clinical significance of these three varieties of lymphocytes are, now coming to the helper T cells first. These are destroyed by the HIV virus which causes acquired immune defense syndrome, and here the person, as they cannot form the antigen presenting cells, they cannot stimulate B lymphocytes, and thus there is no transformation of B lymphocyte to plasma cells, so no antibodies are formed. So when the helper T cells are destroyed, there will be no formation of antibodies. If suppressor T cell activity is increased, they will inhibit formation of T and B lymphocytes, and they cause immune deficiency. And if the suppressor T cell activity is reduced, then more amount of T and B lymphocytes are produced, and they will cause hypersensitivity, and it results in allergic disorders like dermatitis and asthma. Immature cells are known as lymphoblast, and the mature cells are known as lymphocytes. So we will see the histological difference between lymphocytes and lymphoblast. If we take example of any lymphoid organ and the lymphatic follicles present in that organ, then the secondary follicle, which has germinal center or pale staining central area, and dark stained densely packed peripheral zone. So mostly in the peripheral zone of a lymphatic follicle we have the mature small lymphocytes, and in the central part, a pale staining central part is having the lymphoblast. So the lymphocytes are small in size. These are densely packed. The nucleus is larger and they have thin rim of cytoplasm. Mainly lysosomal vesicles are there, and the lymphocytes are arranged along the peripheral or outer zone of the follicle, and when they come in contact with antigen they will start dividing and they form mature lymphocytes. Whereas in the central part of the lymphatic follicle, the immature large sized cells are lymphoblast. They have small nucleus, more amount of cytoplasm is there, and these are pale staining present in the central part of the lymphatic follicle, and the central pale staining part of the lymphatic follicle is known as germinal center. So they enter in the circulation and they form B or T lymphocytes. Again, you should remember that the germinal center is present only in secondary lymphatic follicle. It is not present in primary follicle. And secondary lymphatic follicle is the one which is formed after getting exposed to the antigen. So in the central part, there will be division of cells, immature cells known as lymphoblast. B lymphocytes are originating from bone marrow and they maintain humoral immunity by producing antibodies. Natural killer cells originate from bone marrow and their cytoplasm contains cytolysine, which kills abnormal cells like cancer cells or virus infected cells. These are killed by the natural killer cells, and they destroy antibody bound cells. So this is called antibody dependent cellular cytotoxicity. Antigen-presenting cells include mononuclear phagocytic system. So the example of antigen-presenting cell is macrophages, then the perisinusoidal Kupffer cells in the liver. In the epidermis, we have Langerhans cells. In the spleen and lymph nodes, dendritic cells are there. Thymus, we have the epithelio-reticular cells. So these are all antigen-presenting cells, and they first process the antigen and they displace after digesting the antigen or any microorganism, may it be bacteria or some foreign material. First, it is digested by the lysosomes, and then small peptides of digested antigens are seen over the cell surface by using the MHC 2 molecules, major histocompatibility 2 molecules. So far, we have studied the cells of lymphoid tissue meant for immunity. meant for imparting immunity, may it be T lymphocyte, B lymphocyte, or natural killer cell or antigen presenting cell. These are all related to the immunity. And the last variety of cells of lymphoid system is reticular cells. So they form mainly the structural framework of reticular fibers or reticular cells in lymphoid organs like spleen, lymph node, and thymus. And these histologically, if we see the reticular cells, these are elongated cells and they have long fine cell processes.